board, the Jackie for ALL Foundation. Jackie meant a lot to me, and her loss also affected me, as well as many of my staff, medical, nursing, and other staff. I think a multitude of us are at her wake, and we continue to provide support and to remember her and our contributions on a daily basis. I'd like to just discuss in brief some of the research that's been funded and continues to be funded by the Jackie for ALL Foundation. The formal title of our research is called The Multidimensional Genomic Profiling of Acute Leukemias Characterized by the MLL Gene Rearrangement. Now, many of you may not know what this means, and my hope is at the end of this talk you have a better understanding of what we're doing and how the donations that you make is hopefully going to lead to further improved therapy for this very bad disease. Leukemia is not the most common cancer. It is low on the list of the top 10 cancers. It makes up about 3% of all new cancer diagnoses, but it's a particularly deadly disease with very high mortality rates. Blood cancers in general are normal blood cells that have gone bad, and as you can see here in this picture, there is a normal, what we call a normal stem cell, that gives rise to normal blood cells, and then those cells go and populate your blood and your circulation. What happens in leukemia is that there is a cancerous stem cell, and that particular stem cell goes and grows out of control, spreads throughout your bone marrow in your body, and takes over. Acute lymphocytic leukemia, or ALL, which is what Jackie had, is the most common cancer in children, and you can see here that when you look in the blood, you can actually see these large purplish, bluish cells, and these are actually clearly identifiable as abnormal cells. They don't belong there, and these are actually the cancer cells that we see in the blood of patients that come in with this disease. It's a cancer that, as I mentioned, is characterized by uncontrolled growth of these immature abnormal blood cells. It's the most common cancer in children, so 75% of cases of ALL occur under the age of 6. However, it does occur in up to 20% of acute leukemia diagnoses in adults, as in Jackie's case, particularly young adolescent and young people. Cure rates following standard chemotherapy, as well as bone marrow transplantation, such as Jackie underwent, depend on the particular genetics of the ALL cells. So in some of our ALL patients, we can get cure rates of up to 70, 80, even 90%, but in not all ALLs. Now, in order to understand the research that we do, I'm just going to provide some background on some of the ways that we make a diagnosis and the biology of the blood cancers. One way we can identify blood cancers, obviously, as I've shown you, by looking at the appearance of the cell. The cell does not look normal and obviously looks like a cancer cell. The other way we can identify a blood cancer is to do special marker testing, because the cancer cells express certain things on their surface that we can look for with our instrumentation, and those markers on those cells are not the markers typically expressed by normal cells. The other thing we can look for are alterations in the genetics or the DNA of the cell, and this can be chromosome aberrations and specific gene mutations, which I'll talk about. These are the genetic material that actually makes the cell the cell. In the center of the cell, there's the genes and the DNA and the chromosomes, and that is what determines how that cell will function, and often what determines a leukemia. Just to review, for those people who haven't thought about this since high school biology, as you can see here, this is a typical cell. There's the center of the cell, called the nucleus. And the nucleus of the cell is all the genes and genetic material of the cells. And those genes are they're arranged in these funny-looking X-like structures. Those X-like structures are called the chromosomes. We can see here that each of these chromosomes or X-like structures has a certain look, and if you're a talented cytogeneticist, as we have here, you're able to actually visually look at all of the different chromosomes and see if there's abnormalities in those. Now, on those little X-like structures is actually wound up the DNA, and the DNA is made up of the base pairs. You can see it's unwound there, the A, C, T, and those DNAs make genes, and those genes are what, there's about 30,000 genes that run or regulate how that cell functions. So So there's the DNA arranged in genes. The genes are arranged on the chromosomes. The chromosomes are on the inside of these cells, and they regulate the behavior and what happens to that cell. Cancers have abnormal genetic abnormalities, and you can be reflected either in the DNA, in the genes, in the chromosomes, and the behavior and appearance of those cells. Now, in MLL, which is a mixed lineage leukemia, the gene and the chromosome 11 is a piece of chromosome 11, that chromosome 11 X-like structure is abnormal. And what happens is a piece of that chromosome is exchanged or moved from chromosome 11 to a different chromosome where it doesn't belong. Sometimes that chromosome 11 piece goes to chromosome 4, sometimes it moves to chromosome 19. And when you actually look at the chromosomes, again, if you're very intelligent, you can actually determine what piece of what chromosome doesn't belong, okay? And when cancer is characterized by this particular movement of this particular chromosome 11 piece, tend to do very poorly. So on this graph, if you look on the y-axis, that's the overall survival of patients with leukemia. And on the x-axis is the number of years that they will live. And you can see that if you have acute leukemia that does not have any MLL, that with standard treatment, about 60 or 70% of the infants with this particular leukemia will live. And if you are MLL positive, you will have a worse outcome. Fewer of those patients will be cured by chemotherapy or by transplantation. Because those MLL genes make certain proteins in the cell that contribute to the actual biology of the cancer. Now, as you may or may not know, Jackie's leukemia was characterized by an MLL gene, as I have shown you now. Her particular gene was a movement from chromosome 11 to chromosome 4. So her disease was characterized biologically by this poor genetic marker. So our goal in understanding how these MLL leukemias work and how to cure them and how to treat them and how to best prevent other patients from the same outcome as Jackie is to understand how these leukemias work. And if we can figure out how they work, we don't have to potentially just rely on very high doses of intensive chemotherapy and very toxic drugs to get to where we want, which is cure for the disease. Now, are there any questions up till now? Okay. So our actual project